First of all, thank you to the organizers for the invitation. You heard a lot about Rudy, his work, his life. I will talk more about the work part, less about him. On this occasion, I decided to present basically our last collaboration, which never really kind of had like a proper wrap-up, as you'll see. So Rudy was the person, basically, I did my PhD with him, who got me into electrostatics of viruses. And then, I went for a postdoc soon after I returned. He went to China. So a lot of our collaboration then took place over very, very short emails. And he usually would send me an email. He would say, Anže, I found this interesting. What do you think? And this direction would often be very, very vague. And this is also the story behind this. So there was no clear direction here. It's also, you'll see, it's not, we did a lot of, let's say, theoretical work, and this is not it. This is, I would say, more like explorative bioinformatic analysis. But he was really, really interested in it for, I think, still unknown reasons. And what I'll show today is basically, let's say, a study of the changes in charge on the spike protein of SARS-CoV-2. In hindsight, despite everything, I think the SARS-CoV-2 as a system is unprecedented because, the wealth of data generated during the pandemic is immense. And we got to basically observe in real time how the virus adapted to the new host, how basically changes in its rate of transmission, immune escape, and so on were going on as the pandemic progressed. And estimates kind of show that, any given single nucleotide mutation in the genome has occurred, more than 10,000 times. So in this sense, we can say that the virus has actually explored all possible point mutations. And this basically really allows us to study the influence of different mutations on the virus fitness. This is an example. This is an example of the spike protein here along the amino acid position. Because the spike protein, like David already said, is the one responsible for basically first contact of virus with the cell, with the host cell, with the membrane receptors, is also the one which experienced the most mutations. And this is basically the position of a mutation and basically its relative frequency. And this is the protein itself. It has different domains. The most important one here is the receptor binding domain. There's also the N-terminal domain, and so on and so forth. And basically different mutations in different parts of the spike protein have, different effect on its fitness. What was of interest to Rudy and to me was the fact that the spike protein has a very non-trivial distribution of charged amino acids. Here's basically a top view of the spike protein. This is the side view. And this is kind of like a histogram of the distribution of charged amino acids along the protein. And so you have the stalk part, so the so-called S2 domain, which is predominantly negatively charged. And you have the top part, so basically the one pointing outward, which is predominantly positively charged. However, it has basically a dense negative core, basically core with negative charge. And among the different mutations which occurred in the spike protein, what was basically of interest to us are the changes to and from different charged or ionizable amino acids, because these have the potential to impact electrostatic interactions of the spike protein with its environment. This is an example. So this is the receptor binding domain with the ACE2 receptor, which is kind of the main receptor on the cell to which the spike protein binds to. And what was basically observed relatively early on is that as new variants evolved, especially early on in the pandemic, a lot of changes to charged amino acids occurred in this region. And this basically impacted the binding of the spike protein of the basically this domain with the ACE2 receptor. And you can see here basically the relative binding affinity from the wild type, the reference, with the Delta variant and with the Omicron variant. So this was observed quite, let's say, early on. The thing is that these changes are bound to very specific mutations in this domain. This is another basically example. This is a more detailed view of the electrostatic potential on different variants. And in general, you see that basically with the evolution, basically the charge on the receptor binding motif increases. And I'll get back to this later on. There's more. So it's not only this particular part, but in general, it's thought that, charge on the spike protein influences also non-specific interactions of the spike protein with, for instance, the cellular membrane. Also, what studies have done is they took a look at the binding of the spike protein with different other molecules and receptors on the membrane. And this is an example of a recent study where they took a look at basically, again, the same three variants. So wild type, Delta, and Omicron, and they're binding to heparin, heparan sulfate, and the ACE2 receptor. And you can clearly see here that, something has changed so that basically the electrostatic binding notably increased as new variants have evolved. This is basically, I think, the first paper which kind of pointed this out, and this is also the paper which Rudi sent to me and said, hey, look, this is interesting. It's not necessarily a particularly methodologically good paper, but it gives the correct direction. So basically here you see these are mostly variants of concern as classified at that time with the early pandemic, so basically up until mid-21. And this is basically the change in charge on the entirety of the spike protein. So not only on the receptor binding domain, but on the protein as such. And you can see a trend towards an increase. A later study then checked basically through simulations how this impacts the binding to the ACE2 receptor. And it did basically show that this trend corresponds to improved binding. And there was another also correlation, but this I think is clear that basically change in charge kind of corresponds to, let's say, case fatality, as this was early on in the pandemic, where basically each new variant of concern basically came with increased lethality. But what kind of these studies, let's say, lack is that they mostly focus on the receptor binding domain and its interaction with ACE2. So in a sense, they only look at a small part of the spike protein and basically changes in charge there. And as I said, there, because it's basically the binding is very specific, you don't, a single change in a given amino acid, in a given charge, can have a large impact on how the thing will bind. These studies also almost exclusively looked at just the main variants of concern in the early part of pandemic, so basically up until Omicron. And later on, this kind of basically petered out. And the last thing, there are also many different ways to estimate charge on the spike protein, as you know. I think determining charge on a protein is very difficult. And you can either go like, you can be very detailed, do like ab initio quantum mechanics simulations, but then you're very limited with the size, and you're also very limited at the number of different variants or different proteins you can take a look at. And there's then basically a different way to approach it is you can just look generally, say, you have different like 3 or 4 amino acids which can carry positive charge, 3 which can carry negative charge. You sum them up and you get an estimate of charge, which is a very naive estimate, but you can basically do this among many sequences. this is why what we wanted to do is take basically the wealth of data on the evolution of SARS-CoV-2 and of the spike protein, because basically for every new strain which was detected somewhere in the world, you have sequence data, and just look at the patterns of change in the ionizable amino acids, and basically use that as an estimate to how the charge profile on the spike protein has been changing with the evolution without necessarily saying what the exact charge is. So this is just briefly, in the end, we looked at, more than 2,500 different SARS-CoV strains, which are basically available. And we took a look at the changes in these 6 amino acids. 3 of them, carry positive charge, 3 of them carry negative charge. And we were looking at the changes with respect to the wild type SARS-CoV-2 sequence. And, you can use this as a proxy to determine the total charge, but for the most part, this is not what we were really interested in. And again, what we confirmed is that there's a notable increase in overall positive charge on the spike protein in the early stages of the pandemic. So just to see, here's basically the lineage divergence. So basically how much a given spike protein sequence has diverged from the wild type one. And this corresponds well with basically evolution in time. It jumps around a bit, but it gives you basically the number of mutations from the wild type sequence. And here you have a relative change. So basically this relative change. And you see that basically with these early emerged variants of concern, the first ones which were really noted, you already start to see some notable differences, no? And this becomes basically even more obvious with the emergence of the first Omicron variants. You see that basically you're starting to see an increase in the number of positively charged amino acids and a slight decrease in the amount of negatively charged amino acids. And what's interesting, so, there's a wealth of data for SARS. There's much less available on other coronaviruses, because up until the pandemic, coronaviruses were not really an interesting viral system to study, because at most they cause the common cold. No? So basically there's a limited amount of data on other coronaviruses to work with, but there is some. And so here, here you have the number of the main negative and positively charged amino acids. And this is the basically line of identity. So here you would expect that basically the number of positively and negatively charged amino acids is about the same. So what you see that there are some viruses which are on this line. Some of them are also basically the human coronaviruses, so the common cold coronaviruses. And here you see basically the evolution of SARS. So first of all, you already noticed that it's more, let's say, positively charged, or it has more positively charged amino acids compared to other beta coronaviruses. But you can see that basically with this evolution up to Omicron, it basically, went from having slightly more negatively charged amino acids to having about an equal number of them. And like I said, so this is a proxy, but you can use these numbers to estimate what the total charge on the protein would be. And you can really observe basically this trend, which I showed you initially, we just took the main 6 or 7 variants of concern. You can do this now on basically all the sequence data we have. And you can really see this increasing trend as basically with lineage divergence, so basically with evolution. These are these are the early variants. These are the first seriously evolved variants of concern. And these are basically the early Omicron variants. But one thing you already notice here, that basically this trend of increase seems to have stopped with the early Omicron variants. Because when you look then at variants which have evolved later on, these changes, this notable increase stops and fluctuates actually quite a lot. It never goes back to the original, but you can see that some of them basically go even all the way back to the first evolved variants of concern. The next stage in the evolution were the recombinant variants, so basically variants which took parts of the sequence from different strains. This was another step in the virus evolution, which came with, an increase in viral fitness. And you can see, I would say, a much more varied profile of change. These are the ones here. And to explain the coloring, so basically you can cluster the strains based on their basically profiles of changes in the number of charged amino acids. And this kind of very nicely clusters them basically based on the evolutionary clade. So the very broad profile of the virus evolution corresponds with the changes in the number of ionizable amino acids, which is not something what you would expect. But so this was still at the stage of the pandemic where this was relevant. It corresponds with some other studies which found that basically charge is the dominant factor explaining the lineage divergence. And it means basically that you could use just the changes in basically the charged amino acids to determine the broad division of strains into clades. So you would not need to basically look at the entire profile when determining clades, but you could look only at a specific subset. And the last part, so this is basically where our work with Rudy stopped because of his premature death. So this ended up with an interesting observation for which we can't really offer an explanation, but I think, if somebody would have an idea, you're the audience for it. So this is basically the latest part of the evolution. These are the variants which evolved towards the end of last year. You again see basically a different profile of changes. But the interesting thing is, so if you take a look at the two predominantly positively charged amino acids, lysine and arginine, and you look at their ratio, which is otherwise an important factor in other viruses, you see that basically two jumps occurred, where basically this ratio increases. So basically lysine gets preferred. And with these, basically with the last batch of strains, it increases again. And the interesting thing is basically this does not change the charge. So before I was saying that, in the early pandemic, the positive charge on the spike protein was increasing. But, lysine to arginine or arginine to lysine does not change the total charge. But it still seems to change something because it's clearly, basically, lysine is clearly being preferred over arginine. You can take a look at basically the actual numbers, so now not just at ratios, and you see basically how arginine really drops off, lysine increases, while histidine basically had a peak, but more or less stays the same. And you can also identify where on the spike protein these changes occur. You can see that, for instance, the drop in arginine happens on the N-terminal domain. The S2 is more or less unchanged, whereas the change in lysine predominantly happens on the receptor binding domain. So you can basically also then take this data and identify different regions on the spike protein where the changes are occurring. How much time do I have? So I'll just, because we don't have a conclusion, I'll just briefly go over this. So you can also take some other studies which looked at basically changes in viral fitness from observed mutation counts. So again, you can take evolutionary data, see how often different mutations to and from different amino acids occur, and relate them to changes in viral fitness. And interestingly so, mutations to lysine from arginine, so basically a mutation which otherwise keeps the charge the same, are associated with a decrease in fitness much more often than changes from lysine to arginine. So in this respect, you cannot really replace an arginine with a lysine without basically penalizing viral fitness, even though, as I said, lysine is the one being increased. And this is the same actually on the spike protein and basically on all other coding regions of the genome. But if you take a look at mutations from arginine to any mutation or from any mutation to lysine, you don't really see this change. And again, you can take the data on other coronaviruses, and you see, again, a very interesting thing. So SARS-CoV-2 is a beta coronavirus, which is this part. And this is the initial ratio of lysine to arginine here. And you can see it basically falls somewhere in the middle of the distribution. And this line is basically the maximum ratio achieved, and you can see basically that this ratio has been maximized. So you basically cannot find any other coronavirus which would have such a large ratio of lysine to arginine. And the thing is, so this is where we ended up. We don't have an explanation. There are many. You already heard yesterday that, lysine and arginine are not the same. Even though they both carry the same charge, they have different physicochemical properties which could basically here be relevant for viral fitness. Specifically, for instance, they can influence the stability of the protein and influence basically alternative conformations it takes. There was also a very interesting study which showed that lysine restriction, so diet restriction, can attenuate the severity of the infection, which would kind of be in line that basically if the virus has a preference for lysine, if you restrict it from the diet, you basically reduce its infectivity. And one other possible explanation is that it's actually part of host adaptation. Because eventually, the pandemic is over, but the virus is still here, And in a sense, if the virus wants to stay in the population, it cannot kill it off. So in a sense, a lot of viruses, as they adapt to hosts, they attenuate. So basically, the severity of the infection becomes lesser. And one potential explanation for this particular change is that this is part of the attenuation of the infection as a part of host adaptation. Yeah, with this, I would stop. This is, basically the last work I did with Rudy. It actually ended up unfinished. And what is really peculiar that basically he was really, really interested in this, despite me never really finding out why. Because we never, let's say, this time of the year, when the season of respiratory infections would kind of restart, he would write to me and say, Anže, can you take a look at, what happened over the last year, with the charge on the spike protein, but never really kind of, the entire story never really found a proper end. Yeah, thank you.